Okay, good afternoon, everyone. Am I audible?
Yes, ma'am. Good afternoon, ma'am.
Okay, so I think we can start. We've started the third module, which second module, which is about epidemiology, right? And last time we discussed the journal terms which are used in epidemiology. That what is epidemiology, and what are the terms that are commonly used when we are talking about plant pathogens, right? So when in general we use epidemic and endemic the terms when we are talking about the pathogens or human pathogens, but there are certain terms which are specifically used for when we are talking about the plant pathogen, right? So who is going to tell me what is epidemic called when we are talking about plant pathogen? There were certain term which we talked about that they are what they are called, like epidemic is called for people or on people. So what does when it is called when we are talking about the plants? No one? Epiphytotic was the term used when we discussed about the plant. So epiphytotic is on plants, right? So it was the corresponding terms to the when we are talking about the epidemic. And then in contrast for endemic also there was a term which was called enphytotic. So when we are talking about plant pathogens or when we are talking about the plants, so we are using the terms epiphytotic or enphytotic terms, right? So this means similar like epidemic and endemic. And we also discussed about that how the monocyclic disease epidemic takes place, how the polycyclic disease epidemic takes place, and we discussed the disease triangle. And then in detail, we discussed the factors. We discussed the three factors of a disease triangle in detail, right? What are the host factors, what are the pathogen factors, what are the environmental factors which are important for a particular disease to take place, right? So those things we discussed in detail. So today we are going to talk about again disease forecasting, right? So disease forecasting is a very important part of epidemiological studies. So when we are talking like why we want to study the pathogen or a environmental factor, what are the uses? So one of the uses beside the management is the disease forecasting. That if we are able to predict a particular disease before it is going to happen, then in that case the we can take some steps to to reduce the losses which can be caused by those diseases, right? We are talking about the plant pathogens here, right? So for that matter, the epidemiology, it plays an important role, right? So forecasting of plant diseases, what does it mean? It means we are predicting, right? So forecasting is again a prediction. It is not that 100% this is going to happen. We are just predicting. It is a probability that this disease, the probability of a disease to take place is there, but we are not 100% sure for that happens, right? So forecasting of a plant disease means we are predicting for a occurrence of plant disease in a specified area ahead of time, right? So before it is going to happen, maybe we are predicting for a next week, maybe we are predicting for 5 days later, or maybe for 10 days later. So it is just a prediction, right? So that why this is important, so that suitable control measures can be taken in advance to avoid the losses, as I told you, right? Except viruses, the management of a disease is possible once they are taking place, or there are certain pre-management practices also. For example, fungal diseases, certain fungal sprays are recommended before the disease is happening on the plant. So we can just spray the plant, and after that it reduces the impact of the diseases, or sometimes disease or pathogen they are not able to infect the plant, right? So avoid losses. What steps can be taken in advance to avoid the losses? For that, disease forecasting is important, right? So disease forecasts are predictions of probable outbreaks, right? Probability. I told you it is the probability. We are telling that there is a probability that this disease might take place, right? Or probable outbreaks or increase in the intensity of a particular disease. So it basically this whole process, it require a lot of teamwork, lot of people who are studying that the pattern of that particular disease or that particular pathogen, right? They are already studying so many things simultaneously. And disease forecasting is not a one day process, right? So maybe for 10 years or 5 years, we need to study that pattern of a disease very carefully. What are the environmental factors? What are the pathogen factors? What are the host factors? We need to study. We need to note down them. We need to document those factors. After then only we are able to go for any forecasting program, right? So it can be used as an aid to timely application of any chemical control. And among the first spray warning services to be established for growers, for this is example, this was for the grapevine downy mildew forecasting scheme in France. So this downy mildew and powdery mildew, they are affecting lot of grapevines. Actually, a lot of losses are being caused, right? So among this, they were the like the spray warning one when they should go for a first spray. This was done in Germany and Italy as early as 1920. So disease forecasting methods are available for many plant diseases. This is just a table with a few diseases enlisted here, right? So for grapevine downy mildew in Australia, France, Germany, Greece, Italy, then cucurbit downy mildew in USA, potato late blight. It is India is not included here, but it is in India also. Then apple and pear scab. Again, India is not in the list, but this apple scab disease forecasting program is very well established in India also, right? So a lot of for lot of pathogens, even for a lot of viruses also, there are a lot of disease forecasting programs which are being established, right? So informations which are needed for the forecasting, there are a lot of things we require. It is just not that on the basis of one particular factor it can be done. So we might need a lot of information for a disease forecasting. And how a disease is developing under the influence of several environmental factor, host factor, and the pathogen. So all those are required for accurate forecasting, right? So the factors of epidemic and its components should be known in advance before the forecasting is done. So forecasting is not being done for the diseases which are not established or are not known earlier, or a new diseases. Only forecasting can be done only for the established diseases which are already established, for which we know what kind of environmental factors they require, what kind of a moisture they require, whether they require a rainy season or what is the relative humidity, right? So all those factors are already known. So the informations which are required for the forecasting are again the host factors. So what is the prevalence of susceptible varieties in a given locality? What is the response of a host at different stages of the growth to the activity of the pathogen? How host is responded? For example, some pathogen or some diseases, they impact the seedling stages, whereas some pathogens, they affect the plants when they are grown up, right? So we should know how it is impacting. Then density and distribution of the host in a given locality, right? So dense population, we all know the dense populations, they of a susceptible variety, they definitely invite more pathogens. It is easy for a pathogen to spread in a dense population, right? So growing susceptible varieties in a scattered locations, and that too in a limited area, is a less prone to epiphytotic. So see, we are using here that growing susceptible varieties. So one should think that why we are growing susceptible varieties. These are these basically in those cases where we don't have any resistant varieties available, right? So in those cases, and then those crops are required. These are staple crops. They are staple food crops or vegetables. So we need to grow them anyhow, whether they are susceptible or whether they are. If we don't have any choice, if we don't have any resistant varieties, that means we don't have any choice. But we need to grow those susceptible varieties also. But then we need to make sure, or we need to see that how the infection can be lower down in those susceptible varieties, right? So when we'll study the management practices, so there you'll study that there are a lot of cultural practices, and especially the integrated disease management practices are being followed to reduce the disease, to reduce the pathogen, or to avoid the pathogen, right? So density and distribution of a host in a given locality is again important factor. Then there are pathogen factors, right? So pathogen factors are again numerous, starting from the initial inoculum to the how that inoculum is being spread in the field, right? So amount of a primary or initially inoculum in the air, soil, or a planting material, we should know that what is the how the that inoculum is being dispersed, how it is being dispersed, how the spores are germinated, how the infection is taking place, what is the incubation period of a pathogen, right? So incubation period is time when the first pathogen enter or penetrate the host till the time it shows the first symptom. And as I've told you this earlier also, that incubation period is different for all the pathogens, right? So it is not same. It could be 5 days for some, it could be few hours for some, it could be 15 days for some, right? So we need to check that also. The sporulation on the infected host, and then the how the redispersal or a dissemination of a spore is taking place, what are the perinating stages, what is the inoculum potential and density in the seed, soil, or air. So we need to know all these pathogen factors also. And the important one, environmental factors. So environmental factors are what are the weather variables, what is the temperature, what is the humidity, what is the light intensity, what is the wind velocity. Wind velocity is important because the spores, they are being transported from one part to the other part, right? Through the, if wind is a more in a particular area, so definitely the spores, they'll get traveled to a longer distances, right? So the wind velocity also plays a important role here. Then requirements or conditions for a disease forecasting. So there are some basic requirements which are important, and they should be fulfilled before a successful disease forecast is made. First, right? So how do we decide, right? So how do we decide, and how do we know that for which disease a disease forecasting program is required? Because there are thousands of pathogens, and not for every disease we can go for a disease forecasting program, because it also requires a lot of lot of efforts, lot of human power, lot of economic inputs also, right? So we need to decide that for which pathogen we need to go for a disease forecasting program. So for that we need to look for few things very carefully, right? For example, the disease must cause economically significant damage in terms of a yield loss or a quality, right? So we need to look for the qualitative and quantitative losses, right? Qualitative losses are those which reduces the quality of the crops. उसमें quantity में फर्क नहीं पड़ता, yield उतनी रहती है आपकी, लेकिन quality of the crop is being affected by the pathogen. Quantitative losses are those losses in which the total yield is reduced due to the pathogen, right? So when we talk about the losses, we always talk about the qualitative losses, and we talk about the quantitative losses, right? So damage assessment is essential to develop strategy for controlling a disease. For example, annual estimation of a yield loss, which is caused by a barley powdery mildew in England and Wales, had ranged from 6 to 13%, whereas in potato blight losses were around 28%, right? And even 75% sometimes. So again, then diseases like apple scab and potato common scab, they reduces the quality of the produce, they lower the value of the harvested crops, and it causes considerable financial losses. So when such pathogens are there, which are causes a lot of economic losses to the growers, so then we need to look for some disease forecasting program, or then we can think that, okay, now here we can develop some disease forecasting program, right? And second, the control measures should be available at economically acceptable cost. So economically acceptable cost is important here, that we the management of that particular disease should be available with that at a low cost, because we don't want to put more economic burden on the farmers. So they should be already available for the management of that particular disease, because that's why, and that is when only disease forecasting is useful. If we don't have any management strategies, then there is no point in forecasting any disease. Forecast भी कर दिया तो क्या हुआ, disease आई, उसने affect किया, because we don't have management, right? We don't have any management strategies, तो उसका कोई use नहीं है. So a management ways or a management measures should be available for that particular pathogen or a disease, right? Then disease must vary each season in timing of the first infections and subsequent rate of a progress. If it does not, then there is no need for a forecasting. So it see, this happens. This mostly in every disease is they are not same in every type of a season. There is certain kind of variations. Sometimes the last year the losses could be more, the next year it could be less. So they are never the same. The impact of the diseases is never predicted as the same, right? Then criteria or model which we use for making predictions should be based on investigational work which is carried out in lab and in the field, and then it is tested over a number of years. See the number of years. So disease forecasting is not possible maybe in 1 or 2 span of a year. We need to study that disease for maybe 5 to 10 years to establish its accuracy, and then at number of various locations also. So we need to study those things, right? So growers, they must have sufficient manpowers and equipments to apply control measures when disease warning is given. Long-term warnings or predictions are more useful than short-term. Definitely, if they'll get some time to use those control measures, then only the forecasting is useful. If they get it, just if today they are forecasting maybe tomorrow disease is going to happen or maybe day after tomorrow, then there is very less that farmers can do about it, right? So definitely a forecast after a week or a 10-day time is definitely more useful to the farmers than as compared to the when there's a very less time being given, right? Then methods of a disease forecasting. So it requires a field observations on the pathogen characters. We need to study the weather data. We need to study the different types of the variety of the crops, because it could be there that this variety of a crop is affected more than A variety is affected more than the B. So B is little tolerant or it is less susceptible, right? So all those things, and we need to study. We need correlations here means we need to study the correlation of the disease progression with the environmental factors, right? So it could be the how pathogen is correlated to the environment, how the host factors are correlated to the environmental or weather variables or the disease progression. So correlation between different environmental factors is being studied, right? So forecasting, there are different types of the forecasting. It could be the first one is a forecasting which is based on primary inoculums, right? So presence of a primary inoculum, its density, its viability are determined in the air, soil, or a planting material, right? Primary inoculum is the initial inoculum from where the disease spread will take place. So what is the primary inoculum? What kind of a in what way the pathogen is surviving from the previous season, right? So how much amount that inoculum is present. So occurrence of a viable spores or in the air, they can be. So there are different methods for this. There are certain spore traps which were designed to see that how much spores are present in the air, or there are certain air trapping devices which are called as a spore traps, right? So, and in case of a soil-borne pathogens, the primary inoculum in the field can be determined by the monoculture methods also. So there's example here, the presence of a loose smut of a wheat or ergot of pearl meat and viral disease of potato can be detected in the seed lots by different. So those in those cases, right, where the starting material or that material that we are using in the planting for the new crops can be the source of infection, right? So in those methods we can go for a several testing, right? So you remember last year we discussed that viral disease management, there was one important step, that certification of a seed material or a planting material. What does that mean? That means we need to check the starting material for the presence of the pathogen. So similarly, we can check the seeds and all those lots by the different methods, whether they are some kind of a infection already there or not, right? So seed testing method can be used to determine a potential disease incidence, and it can enable decision to be made on the need for chemical. So if we find some kind of a spores or some fungal spores or a bacterial spores or infection in those seeds, so certain seed treatment methods are available, right? So we can treat those seeds before we sow them into the field, right? simpler methods are a hot water seed treatment. So at a certain hot water, you dip those seeds, you take out those seeds. So because higher temperature also kills the fungi or other kind of a spores, or there are certain fungicides are available for the seed treatment specifically. So we can do that also, right? So extent of many virus diseases. Here we are talking about the viral diseases. It is depend upon the severity of the preceding winter, which affects the size of vector population in the growing season. So definitely, since viral pathogens, they are spread by the vectors, so vector population in a particular area can give us idea that if disease is going to happen or not, right? Or if we can manage that vector population, so definitely we can reduce the disease impact somewhere, right? Then forecasting, which is based on weather conditions, right? So weather conditions, the weather variables such as rainfall or relative humidity or a temperature, right? During the crop season and the intercrop season, they are measured. So during that time, we take those weather data is being recorded basically, right? क्या temperature था, क्या relative humidity थी, rainfall कितनी थी, everything is being recorded. So weather conditions above the crop and at the soil. So this is different here because weather conditions, temperatures can, because there's a crop canopy, right? So above that, if the plants are very dense or there are taller plants, so we'll see the temperature near the ground level will be lesser than the temperature at the above the crop, right? So those variables are also being recorded. Then forecasting based on correlative information. So weather data of several years are collected and it is correlated with the intensity of the disease, right? So weather data हमको कहीं पे भी weather forecasting area से मिल जाता है. Usually everywhere they are present, right? So we'll collect that weather data and we'll correlate it with the intensity of the disease. We'll see whether with the change in weather how disease is behaving, how pathogen is in, whether it is increasing or it is decreasing, what is the impact of weather variables on the pathogen, right? So the data are compared and then forecasting of the disease is done. So forecasting criteria which are being developed from the comparison of disease observation with standard meteorological data. Meteorological data means weather data, which have been provided by number of like septoria leaf blotch of a wheat, fire blight of an apple, and barley powdery mildew. So these kind of correlation data have been developed for these kind of a pathogen. Then these days we have a computer-based disease forecasting. So all we need is to there are certain programs which are being made, and all we need is to feed data in that computer. The older data is already available. We'll just feed the present data which is prevailing, and by calculating the kind of the disease which has been prevailed in the previous years on the basis of those weather conditions or other factors, these programs, they are going to calculate the present condition and they are going to forecast, right? So forecasting of a disease is made by use of a computer. This system gives the result quickly, and there are a lot of computer-based programs throughout the world. It is not only in the US, there are a lot of programs which are being developed. One such is a blight cast for a potato late blight, which was being developed in the US. So again, these are the certain programs which are being developed for the pathogens are being used. Early and late leaf spots of a groundnut. So there was a technique which was developed for forecasting early and late leaf spots of groundnuts in the USA. So they found that when the groundnut foliage, it remained wet for a period greater than or equal to 10 hours, and the minimum temperature is around 21 degrees Celsius or higher for 2 days or a night, the disease development is forecasted. So if such weather conditions they prevail for 2 days or 2 nights, they are going to forecast the disease, because now they have noticed from the previous year surveys that disease is going to take place. So a computer program has been developed in the USA, and the data on hours for day with relative humidity of 95% and above, and the minimum temperature during the relative humidity observation for a period of 5 days, they are being calculated. So they calculate the temperature and the relative humidity index for each of the 5 days. So if this index for days 4 and 5 are summed, so if the total index exceed the 4, the disease is being forecasted. If the disease index is 3 or less, that means no disease will take place. So this is how the calculations are being done. So they do, again, there's a late blight of potato. So in they have a forecasting system for late blight of a potato, Phytophthora infestans. And we in India also, we have this late blight forecasting program, which is called as a it is called as a just a moment, it is Jhulsa cast. Jhulsa cast. So Jhulsa means like a burned, right? It's a name given in Hindi basically. So this Jhulsa cast program is disease forecasting program for potato, is being used in Punjab and those sides where the potato is being grown. So Jhulsa cast, Jhulsa means, they've given a Hindi name. Jhulsa means like a blight, means like burned, right? It gives a burned appearance. So on that they named it as a Jhulsa cast, right? So the initial appearance of late blight is forecasted 7 to 14 days after the occurrence of 10 consecutive blight favorable days, right? So favorable days means the temperature, humidity was favorable for the pathogen. So a day is considered to be blight favorable when 5-day average temperature is around 25.5 degrees Celsius and the total rainfall for the last 10 days is more than 3.0 centimeters. So this is how they forecast it. Then the computerized version, which is called Blightcast, is developed in USA for forecasting of potato late blight. So they have a it's a computer-based program basically on which they have made this Blightcast program. So when a farmer desire a Blightcast, so when they want to know whether this disease is going to happen, they'll call the Blightcast operator and they'll report the most recently recorded environmental data to them, right? So the operator calls for the Blightcast programs in the computer, right? So they have a computers, they'll feed that data into the computer, and within a fraction of a second, the computer will analyze the data and series of forecast and spray recommendation to the operator who relays it to the farmer. So this is how it is being done in a computerized manner also, or through using computer-based programs. So the entire operation can be completed during standard 3 minutes telephone call. So it is why this is easy, because see, farmers, we know they don't have computer, they are not going to do it themselves. So all they need is to just do a phone call to the operator who is dealing with this forecast program, and it is being done, right? The system makes one of the 4 recommendations, like they'll tell whether there is no spray, whether there's a late blight warning, or there's a 7 days schedule for 5 days spray schedule, right? So this is how they're going to tell that how they are going to need spray to prevent the particular disease, right? So again, right. So again, in West Germany, Phytoprog is the program which is used for this, and it is again based on measurements of temperature, relative humidity, and rainfall. This is again the one program. Then we have a blister blight of a tea. So a system for predicting epidemics of blister blight of a tea has been developed, and it is based on the number of the spores which are present in the air in the tea plantation, right? And the leaf surface wetness, for how much time the leaf surface is wet. The duration of sunshine is negatively correlated with the duration of the surface wetness here. Then we have a southern corn blight, EPIME, which is a system of forecasting for southern corn blight. Again, it is a conceptual model based on a conceptual computerized model. Then this is rice blast in India. So forecasting rice blast, which is caused by pyricularia or oryzae, is done by correlative information method here. Correlative again means we are correlating the weather variables, different weather variables, right? So it is again predicted on the basis of minimum night temperature, which should be 20 to 26 degrees Celsius. And we are comparing it with the high relative humidity. It should be 90% or above, right? So computer-based forecasting system has been developed for rice blast in India. Then we have a wheat stem rust. Rust in a wheat is a big problem. Yellow rust, brown rust, or a black rust. It causes total losses in the wheat, right? So forecasting wheat stem rust epidemic is done by again analyzing the rain samples, which gives what kind of inoculum or how much inoculum is present in the air, right? So it has been observed that primary inoculum comes from South India to the plains of Central and North India. it is usually through the wind. See, it can go that far also by the wind velocity. And then we have a brown stripe downy mildew of the corn. So the forecasting of a brown stripe downy mildew of a corn, which is restricted to India, is done on the basis of average rainfall and the temperature. So we study what is the rainfall and what is the temperature. Then we have a certain spore trapping techniques which are required for the forecasting, right? So there are spore traps which have been widely used to complete the disease with weather conditions. So what kind of a spore traps are there? I'll just tell you. So spore trapping is useful for understanding epidemiology of a disease and behavior of the pathogen. So this again helps in developing models on the dispersal of pathogens or on epidemiology of the disease, and then we can go for the management, right? So methodology for spore trapping again depend upon the biology of the pathogen, for the infection forecasting, for the spore dispersal gradients, and for the management of the disease, right? So there are certain methods which are available for to trap the spore. So they sometimes they use cylindrical rods or microscopic glass slides, right? So it helps to gather data on the spore arrival in the particular locality. So the the microscopic on the microscopic slide they put some grease, right, so that it becomes sticky. So here the quantitative estimation is not possible as number of spores collected are very low here, right? Then there was this HIST volumetric spore trap, which was given in 1952, right? So again, the number of spores per unit volume of air at any given time can thus be calculated by this method. Then there was a rotorod sampler or rotorod spore trap, which was given by Sutton and Jones in 1976, right? So it has some U-shaped rod which is attached at its midpoint to the shaft of the small battery which operates with the electric motor, right? So from the surface of the strip and the speed of rotation, the volume of air samples can be calculated by this method. Then we have a Baudelin slit sampler. Here the air is sucked in a chamber by vacuum pump, which strikes the rotating petri dish, which contains the agar medium, right? So agar medium retains the spores which are sucked in the air, and the concentration of a viable spores can be calculated by this method. Then fifth one is we have Burkhardt 7-day volumetric spore trap method. And again, this device records spores in the air which are drawn by a pump on 7-day basis. So again, this is some method. Then we have a jet spore trap method also available. So here the above methods which we discussed where the viability cannot be done, but to overcome this, living plants have been used as a spore traps. So a spore trap is where spores are impacted in air jet into a column of a still air through which they fall to settle on the leaf segments and exposed at the base of the chamber. So in this trap, suitable cultivars of host plants can be employed to determine the number of the viable spores also. So these are the certain methods, or today we have discussed the examples where the disease forecasting system have been developed and have been used in India and abroad. Beside this also there are there's EpiVen for apple scab, which was very widely being used in India also. And as I told you, the late blight of potato forecasting system has been used in India also, Julsa cast, right? So similarly, we have number of forecasting systems which are being used. So we discussed basically about the fungal or bacterial, mostly on the disease forecasting, they are available for the fungal or bacterial pathogens. But yes, there are certain examples where we'll see the disease forecasting is available for the viral pathogens also. So that is again one lecture I'll just give you on that also, that how disease forecasting with examples in which cases the disease forecasting in viral pathogen is being done. So there'll be one more lecture on the disease forecasting for viral pathogens. Yes, there are very not as many as examples which we can see in fungal pathogens, but yes, in sometimes in some diseases, the viral diseases, the losses caused are more, and we need to develop the certain forecasting procedures for the viral pathogens also. So in next chapter we'll try to discuss that. So that's all for today, and if you think that there is something you need more clarification, it's open for discussion to everyone. Okay, students. So just go through this also. Disease forecasting is a very important part of epidemiology, and most see in disease forecasting also we need to study only those things of a disease triangle that we studied last time. So things are overlapping here also. Just go through the chapter, go through this also, and I don't think so you'll find any difficulty. But still, if you feel that it is difficult and you need to study some more examples on this so we can go for see the this disease forecasting is totally mathematical modeling, which I'm not including here, and maybe I'm even not the right person to discuss that. So when we talk about a disease forecasting, there are a lot of mathematical models which are available, lot of calculations are being done, and then because when we are talking about the correlation also, so correlation, to find out those correlation also we use the biostatistical methods or formulas for those also. So we are not going to discuss that here because that is again of a very higher level. So I'm just trying to give you the overview. What is disease forecasting? What are the basic requirement for a disease forecast program? How do we decide? For example, we take a hypothetical solution that you are given an option that you have to you have to make a disease forecasting program for one particular pathogen in particular area. So how you'll select that, right? How you'll select that? So those examples or those basic points I've given you, that on what basis you are going to decide whether you're going to develop a disease forecasting program, or whether a plant pathologist or a people who are involved, how they are going to decide whether they want a disease forecasting program for a particular disease, right? So for that you need to study number of factors. What are the quantitative and qualitative losses? What are the disease management practices available? How many years that disease have been studied already? Whether we have any kind of a data which tells us about the correlation of the weather variables and the disease progression in a field, right? So all those factors we need to study, and if we have all those factors, then only we can go for a disease forecasting. So first important thing is the disease losses, quantitative, qualitative losses. Second important thing is the management practices that we have available. Because if we don't, as I told you earlier, if we don't have any management practices, then there is no use for a disease forecasting program. So if management practices at an economic cost, right? They should not be very costly for the farmers. They should be available, right? And then we discussed the examples of a few diseases where the disease forecasting is available in few pathogens which are important and which are causing a lot of losses. So in those, the disease forecasting is available. Just give me 1 second. There's one call. Hello. Okay, so disease forecasting programs are very important for the management of a particular disease and to reduce the losses which can be caused by a particular disease if they are not being forecasted. For example, in fungal and bacterial diseases, we can definitely take some steps, or in viral diseases also, because viral diseases are dependent upon the vectors. So if we can manage the vector, we can reduce the disease. Similarly, in fungal and bacterial pathogens, there are a lot of spraying programs or chemical control programs are available. If we take those steps before disease is taking place and it is being forecasted earlier, so we can reduce the losses which are being caused by the pathogen if they are not being forecasted, right? So this is how the disease forecasting programs are useful. They are useful in reducing the losses which are caused by a particular pathogen and giving some time to the farmers to take some steps to manage that particular disease, right? Okay, students. So I guess that's all. I'll stop it here, and maybe next lecture will take maybe day after tomorrow. Tomorrow is a holiday, right? So we can just again if you have some issues. See, after every lecture I tell you that whenever you go through the lecture and you find that this thing is not clear, just underline that or tick that, and in the next class you can always ask me to discuss that thing again, and we can always discuss whenever next class is happening, right? So just make sure that you go through these things carefully, right? Okay, so for attendance, please switch on your cameras. Yes, Adish Mishra, Athoiba,
Present, ma'am.
Ayushi,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi,
Present, ma'am.
Ishita,
Present, ma'am.
Janvi,
Present, ma'am.
Kshitij,
Present, ma'am.
Namrata,
Present, ma'am.
Nehal,
Present,
Rupal, Rupal,
Present, ma'am.
Sahiba,
Present, ma'am.
Shakti,
Present, ma'am.
Shivani, Tanvi,
Present, ma'am.
Siddhev, Vivek, Yukta, Yuvraj.
Present, ma'am. Present, ma'am.
युवराज युवराज की आवाज नहीं सुनाई दे रही है. Okay, Yuvraj, fine, there you are. Okay, students. So Thursday we have a practical, so we are going to have this presentation also, and maybe one small lecture we can schedule that day, right? So I'll see you day after tomorrow. Thank you so much. Bye bye.
Thank you, ma'am.
Thank you.
Thank you, ma'am.